Okay, good morning, everyone. So did you guys have a good weekend? Oh, good. I had a good sports weekend. Not only did the Warriors win, but the Lakers lost, so I was pretty happy about that. I've disliked the Lakers since I was about 2, I think, so. Secondly, more importantly, one of my daughter's lacrosse teams won both of their games over the weekends. So one of my 10-year-olds, so that was even more fun. So I want to just review a couple things, and at the very end of the lecture, I'm going to go over again the difference between insulin receptor and the general category of receptor action, as I ended on Friday. So first off, steroid hormone action, it's all defined by, in terms of specificity, 3 the critical binding domains that we talked about. The steroid binding domain, the DNA binding domain, and the transcription factor. They all bind on the promoter of a target gene to regulate gene transcription. And the binding sequences are in the promoter itself. And we talked about a gel shift assay that can define experimentally what these binding sites are. And what you follow is, for the glucocorticoid receptor, they have a glucocorticoid regulated element, which is different than what the estrogen receptor binds to, which is the estrogen regulated element. Again, we talked about this. And you base it on size differences. And the surprise in the field was, it wasn't so surprising that the glucocorticoid receptor bound to a glucocorticoid response element and an estrogen receptor bound to an estrogen response element. The surprise was progesterone receptor, mineralocorticoid receptor, and androgen receptor, steroids involved in pregnancy, male reproduction, sodium metabolism, bind to the same DNA site as glucocorticoids, which is your stress steroid. So it says the binding domains of the steroid binding domain and the transcription factor, the protein binding domains, are very important for specificity, because in the case of these 4 steroid receptors, they bind to the same DNA. So it tells you how important everything is. You need all 3 binding domains to have this specificity. So it's a very unusual example. It was not the prediction in the field at the time when they did the experiments. And the gel shift assay is what they used to show this. Okay? Any questions on gel shift assays? Again, that was a long time ago on Friday. Okay? And then to show you how important these 3 domains are, we talked about a syndrome called AIS, androgen insensitivity syndrome. We have genetic males look female. The external genitalia look female. In fact, when they're born, you think it's a girl. But it's actually a genetic male. And there's defects in the androgen receptor, because you need a shot of androgen and respond to it during fetal development to look like a male on the outside. So the default pathway is female looking. And the defects are in androgen receptor expression, so it's a promoter mutation. In addition, you have defects as predicted by these 3 important areas of specificity. The vast majority of the mutations line up with the steroid binding domain, the DNA binding domain, or the protein binding domain of the androgen receptor. So that shows from a clinical manifestation of a mutations that these 3 binding domains are incredibly important for steroid receptor specificity. Any questions on that? I bring that up because it's a very nice way of showing that one base pair mutation can truly change the phenotype of an individual. And not so much the physiology, but more the phenotype in this case. A little bit of the physiology. Okay, so now we talked about glucose homeostasis. For those of you who had breakfast this morning, as we talked about, high glucose kicks on the beta cells of the pancreas to secrete insulin. Insulin binds to receptor in the liver. And you bring in glucose, you store it as glycogen, and you use it. And we'll talk a little bit more about that aspect in diabetes in a second. Low glucose, alpha islet cells produce glucagon. Glucagon binds to its receptors in the liver. And glycogen gets taken down into glucose, secreted, and you produce more glucose. And this is our starting point for discussing 2 general classes of receptors. Because if you know about insulin receptor and glucagon receptor, you know a lot about cell surface receptors. And these bind receptors on the surface themselves. So I ended with a discussion of insulin receptor, and I just want to clarify one particular important issue. The general class of receptors, these are single transmembrane receptors, and they have a tyrosine kinase on the intracellular domain. When the hormone binds, these guys dimerize. It is the physical contact of the dimerization that activates the tyrosine kinase on the receptor. Okay, so what does every self-respecting enzyme must have? A substrate, right. In this case, the substrate is the receptor itself. So it cross-phosphorylates the two subunits and allows that as landing sites for signaling molecules. And that initiates the response. A lot of these signaling molecules are themselves enzymes, bridging compounds, and connect into very large signal transduction pathways, none of which you need to know particularly about for this. You'll get that in upper divisional classes. Okay? Any questions on the general class? Now, insulin receptor is part of this general class, but it has two very important distinctions. One is the insulin receptor is already in a dimer. So when the hormone comes in this case insulin, you get clusters of dimers. Not just dimers, but clusters of dimers. But the net effect is the same. The physical contact of the receptors with themselves causes the tyrosine kinase to activate. And again, what does every self-respecting enzyme must have? Substrate. So in this case, you have two substrates. One substrate is the insulin receptor itself, the other subunit, just like the general category of the enzyme of these receptors. Secondly, it has another set of substrates called IRSs, not Internal Revenue Service. I know your parents are worried about that this week. But it's insulin receptor substrate 1. There's about 5 or 6 of them. You only need to know that they're insulin receptor substrates. IRS1 is the more important one. This also comes up into the tyrosine kinase, and it gets phosphorylated on tyrosine residues. That's another distinction of insulin receptor to most of the other class of these general class receptors. Okay? And signaling molecules, just like with the general class, bind to the receptor itself and bind to IRS1, kicks on a response. So view these receptor substrates as an expanded platform of the receptor. So instead of being able to bind 5 or 6 signaling molecules with this expanded platform, now you can bind and activate maybe 20, 25 different signaling molecules. And as you might expect, insulin has a pretty broad range of responses that are dictated by who binds to the receptor and to IRS1. Okay? Any questions on that? Okay, good. Now, people with defects in regulating glucose in the insulin response system are diabetic, and there's 2 general classes of diabetes. One is type 1, and type 1 diabetes results from an autoimmune disease where the beta islet cells are destroyed. So if you destroy a beta islet cell, what do you think the defect is? If you destroy a beta islet cell, in the presence of high levels of glucose, what won't happen? Produce insulin, that's right. So the defect in type 1 diabetes is a complete loss of insulin. And this particular form of diabetes represents about 10% of the diabetic patients, and these are patients that you can treat them by giving them extra insulin. Okay? In contrast, if you look at the pathway of insulin receptor, going from the hormone all the way down to these signaling molecules, if you had a defect at any point in this pathway, what would you predict about your ability to respond to insulin? Would it be the same, better, or worse? A lot worse, right? And that represents a defect in that pathway, the other kind of diabetes called type 2, which is a manifestation of lifestyle, combination of lifestyle and genetic factors. And this tends to have loss, actually, it can result in lower insulin levels, but not a complete loss. Alterations and defects in the receptor. And importantly, which is really clearly recognized, is defects in the signaling pathway. Many people with a type 2 diabetes, which is adult onset diabetes, really are defects, not so much defects, but changes in these downstream receptor events, phosphorylation, signaling molecules, and further downstream. And that's why you can partially control this by diet and exercise, because there's a very complicated interaction of all these signaling pathways with your overall physiological state. Okay? Now, what if an individual had a tyrosine kinase in its insulin receptor that was completely dead, had a mutation in the active site, right? Every self-respecting enzyme not only has a substrate, but has an active site, right? What if you had a point mutation in the receptor's kinase that killed activity? What would you predict about the ability of insulin receptor signaling to work? Would it be better, worse, or the same? It'd be a lot worse. Because everything, if the kinase is dead, you could not phosphorylate the receptor or the substrate. And if you can't do that, the signaling molecules cannot bind. And if you cannot do that, you've got no response. Okay? And they've isolated actually several type 2 diabetic patients. There's a few out there where the kinase, there's a mutation right in the active site of the protein kinase, of the tyrosine kinase of the insulin receptor. So it's something you do see clinically. Okay, any questions on that? And obviously, if 10% of diabetic patients are type 1, about 90% are type 2. And this is the majority of people out there. And those of you going into medicine will probably see this quite a bit because it's adult onset. So people will just get below that threshold of glucose regulation where they become diabetic over time. Okay, so if you see your parents, tell them, eat well and exercise, because it will hold this off a little bit. Okay? Questions? Now, one thing that's not in the reader is another class of receptors that I will talk about the receptors themselves, but I just want to give you a sense of the mechanism. And this will be very, very brief here. These are transmembrane receptors without an enzyme, but they bind to an enzyme. And some of the hormones we'll talk about that do this, we've already partially talked about it. Growth hormone, prolactin, which we'll talk about briefly during lactation, probably by Friday, and interleukins, which are involved in the immune system. And these, in a general way, we're not going to go into too many details. And there's similarities to the mechanism. So here's the receptor sitting in the plasma membrane, does not have any enzyme in it, but nearby is a tyrosine kinase. The hormone binds, and you cause a dimerization of the receptors, but instead of activating the enzyme, what happens is it creates a landing site or a higher affinity for the tyrosine kinase, which is now active. Whereas here, the tyrosine kinase is inactive. Again, what does every self-respecting enzyme must have? Substrate, right? So in this case, some other substrate with a tyrosine residue comes up to this tyrosine kinase in the presence of ATP. Call this Y. And it gets phosphorylated on the tyrosine residue and mediates a response. In the case of prolactin, this molecule is a transcription factor. So here's a case, again, you don't have to know this. This transcription factor comes up to the membrane, gets phosphorylated by this tyrosine kinase, then shoots back down to the nucleus and regulates genes. In the case of prolactin, one of the genes it regulates is casein, which is your milk gene, your milk protein. And prolactin is, as you can get from the name, prolactin, involved in lactation. So it all kind of ties together from that standpoint. Note the differences generally, that the enzyme here is not in the receptor. The enzyme is next to the receptor. And the dimerization activates a protein interaction that then activates the enzyme itself. Note the difference between that and this general category of enzymes. Okay, any questions on this? Good. Now, how many of you have not had breakfast this morning? I know I asked you that several times. Notice there's less. Each time I'm embarrassing you guys a little bit. Okay, that's good. However, you guys undergo a completely different receptor signaling pathway because you were probably a little hungry. Probably some of you still a little sleepy and stuff, but you're probably a little hungry. And so instead of needing to bring glucose into the liver, you need to generate glucose from your liver. And that involves the glucagon system, is another receptor. So glucagon works through a completely different kind of receptor system. This works through a 7-pass transmembrane receptor. So 7-pass means it goes through the membrane 7 times. And it interacts with a G protein. Here's the receptor. It interacts with a G protein that in its inactive state is bound to GDP. So this circle, this pseudo circle here is the protein, and it has this nucleotide GDP bound to it. Okay? So what happens when the hormone binds, when glucagon binds to its receptor in the liver? Just call it hormone here, a little easier. The hormone binds, and what you have is an exchange. GDP comes off the G protein, GTP goes on. So the hormone changes the conformation in a way that this G protein kicks off the GDP, kicks on GTP. So it's an exchange reaction. Okay, you can't just synthesize GTP at the membrane. Right? You guys know that, right? All in the oxidative phosphorylation, high energy bonds, mitochondria, right? That all has to happen there. So as a result, this G protein, I'll leave this on the board up here. This G protein is now active. And what happens is the G protein then that's bound to GTP moves off the receptor. Let me just put this like this. Moves off the receptor, and this G protein binds and activates other enzymes. One of the key ones that it activates is adenylate cyclase. Which converts, we'll talk about the downstream pathways in a second here, converts ATP into cyclic AMP. And I'm not going to test you on the structures. It's in the reader, or it should be in the book. But it's the phosphate group is, you have one phosphate group that kind of forms a circular kind of structure, a ring type structure. This is now active. This adenylate cyclase is now active. But in any hormone response, it's not just turning it on. You have to turn off a hormone response, right? Remember, on all these big negative feedback homeostatic systems, within an acceptable window, you have to go up and down, right? So how does one turn this off? You turn this off by regulating this G protein. So what happens is the G protein actually has an enzyme in it. This G protein is an enzyme, and it has a GTPase in its structure. So as you can imagine, if you ever see the word ASE, or the ending, it generally means cleavage. And you'll see that in digestion, proteases, lipases, and so forth. But this is a GTPase. So what happens is the GTPase starts working as soon as the G protein binds to the adenylate cyclase, and you kick off that last phosphate group, right? GTP is 3 phosphate groups in a ribose, right? You've had that earlier. You kick off the last one, the gamma phosphate group, and you end up with GDP bound to the G protein. Everybody see that? This is the off switch. The exchange is the on switch. The off switch is the GTPase. And it's the relative activities of the exchange and the GTPase dictate whether you keep adenylate cyclase active or not. So GTP-bound form on, GDP-bound form off. This then, the GDP-bound form, binds to the receptor and waits for the next hormone to come in. Everybody see that? Any questions? So basically the hormone, you're hungry, you've produced glucagon, you cause a GDP-GTP exchange, the GTP protein binds to adenylate cyclase, you kick on adenylate cyclase, we'll talk about what happens downstream of that in a second, and you turn this off by this GTPase that cleaves the gamma phosphate group. GDP comes back on the enzyme, it's inactive, waits for the next hormone. As I mentioned, you have to keep homeostasis, you have to be, when it's too low, you go back up, right? You kind of keep things within an acceptable window. There are disease states that mess up this pathway. One of them is cholera. You guys all heard of cholera, right? It's a pretty big-time disease in the world. What cholera does is, I don't want to get into the details, but what cholera toxin does, it covalently modifies the G protein in a way that it keeps it on. So with cholera, you always have the GTP, G protein, in this GTP-like state. And as a result, what would you predict about adenylate cyclase if the G protein is always in the GTP-like state? It would always be on. That's right. And if you have too much cyclic AMP, you have a lot of problems. If you never turn that back down, you never dial it back down. One of them is, for example, dehydration is a big issue in cholera. That's usually what people die of, actually, is dehydration. This alters that system pretty dramatically. And that is also why it's so hard to treat it right away, because the cholera toxin gives you a covalent modification. So you just can't turn it on and off. You've got to actually turn over, degrade the G protein, and make new G proteins in the absence of the cholera toxin. So that's why even when you give these people very lots of clean water, it takes them weeks and weeks to fully recover from this thing. It's one of the reasons, because it's a covalent modification. So what you're doing is you're changing in a more permanent way the signal transduction system. Okay? Another one, I'm not going to talk about how it works, but whooping cough also affects aspects of this system. Okay, pertussis toxin, which is whooping cough. So those of you getting into medicine will see this pretty directly. And of course, there's a lot of medicines that are clinically directed at this pathway, as you can imagine. Okay, questions on that? So on these signaling pathways, you don't want to leave them on. Normally, you want to be able to turn it on when you want, turn it off when you want, turn it on when you're off, turn it off. Anytime you keep it off permanently or keep it on permanently, not a good thing physiologically. Okay, questions? Yes, question.
Okay, so you said the exchange activates the release of glucagon, and then you said
No. Glucagon binding to the receptor changes the conformation in a way that this G protein now exchanges GTP for GDP. And the turn off, that's the on. The exchange is the on. Activates adenylate cyclase, and then to turn this off, the G protein itself has a GTPase. So this is the off switch. Soon as the GTPase cleaves off this phosphate group to give you GDP, at that split moment, it no longer can bind to adenylate cyclase, but instead it binds to the receptor. Okay, so you're altering receptor adenylate cyclase interactions by whether you have GDP or GTP on the G protein. Does everybody see that? It's all protein-protein interactions. If you are your hormones, you're certainly regulated by protein-protein interactions. Yes, there was a question here. One. Is there a genetic disease based on this process? Yes. There's multiple ones. Because you can see any of these signal transduction pathways, a defect in any one component or a change in expression can alter how you respond to your external environment. In this case, glucose homeostasis, which is really important. Okay? Now, what goes on downstream is also a lot. As I mentioned about cyclic AMP, what happens is, I'll leave this up here for now. There's a protein kinase called protein kinase A. So the question is, what does cyclic AMP do? Protein kinase A, or pKa, is a serine threonine kinase. Okay, what do you think its substrates have if it's a serine threonine kinase? Or what does it do to its substrates? Phosphorylates them at the serine or threonine residues, right? Unlike a tyrosine kinase that phosphorylates at a tyrosine, this guy phosphorylates you at a serine threonine kinase. It has two subunits, a regulatory subunit. This is sitting in the cytoplasm, and a catalytic subunit. Call this reg and cat. Cyclic AMP comes by, binds to the regulatory subunit. And I'm not worried about the stoichiometry here, but it's a binding which releases the catalytic subunit from the regulatory subunit. This is now an active kinase, an active serine threonine kinase. Okay, does everybody see that? Cyclic AMP comes in, binds to the regulatory subunit, releases it, alters that protein-protein interaction, yet another protein-protein interaction. These are each proteins. They come apart, and this catalytic subunit is now active. Okay? And this catalytic subunit that is now active can run amok in the cell. It's one of the most promiscuous kinases known. It sees any serine threonine, it wants to phosphorylate it. Okay? And it has a lot of effects in the cytoplasm. Now, when you produce glucagon, are you hungry or are you well fed? You're hungry, right? Because you want to produce glucose. One of the things it does is it kicks in the cytoplasm, causes a pathway, it's more than one step, causes glycogen to eventually become converted to glucose, broken down to glucose, and kicked out the cell. That's one way in which you produce glucose. This catalytic subunit can also go into the nucleus. Can you guys, besides histones and other kinds of proteins, can you name one thing we've talked about in the last 4 lectures that's in the nucleus? A class of proteins? A transcription factor, right. There's transcription factors in the nucleus. One of the things it does in the nucleus is, don't need to know the name of the transcription factor, phosphorylates the transcription factor, and this transcription factor, do this over here, sits down on genes that are involved in glucose production, and these sit down on gluconeogenic genes. So they produce glucose. So when you're hungry, you have a cytoplasmic effect that you break glycogen down to glucose, and you have a nuclear effect that you produce genes, increase the synthesis of genes that produce more glucose, which is what you want when you haven't had breakfast. Okay? So those of you who had breakfast already, about 11:30, this is what's going on. Okay? Questions? Okay, so cyclic AMP is, in fact, considered your classic secondary messenger. And just a real brief description of secondary messengers, or one aspect of it. I'll put this up here so you can still have it. If you look at the overall pathway, you can say the hormone comes in through a bunch of steps. You make cyclic AMP in the cell. And then you have a bunch of phosphorylation events, and then you have your response. Very general way of thinking about this. A classic secondary messenger means the hormone causes the production of the secondary messenger, and the secondary messenger is a singular responsible compound for the response. And you can prove that a couple ways. One way is you can take a cell and you can give it a form of cyclic AMP that goes through the plasma membrane. If this is a true secondary messenger, would you predict that this response should occur in the absence of the hormone? If this is the singular responsible molecule. You'd predict that, right? Everybody see that? And that's exactly what you see. You take a cell without the hormone. You add cyclic AMP, and you get the response. You absolutely can do that. Secondly, this phosphorylation is due to protein kinase A, right? It runs amok in the cell and gives you a whole bunch of different responses. What if you knocked out pKa? And this was absolutely critically important for your cyclic AMP and your hormone response. If you knocked this out, what would you predict about this response if, in fact, it was really important? You shouldn't see it, right? Does everybody see that? And then, in fact, that's exactly what you see. You knock out protein kinase A, you can add all the hormone you want, give all the cyclic AMP you want, but you get no response. That shows I just summarized to you about maybe 20 years of experiments. Okay? That cyclic AMP is responsible for this glucagon response, and it must activate protein kinase A in order to see the response itself. So it's a sequence of events. You're hungry, you get a hormone, binds the receptor, GTP, GDP exchange, activate adenylate cyclase, produce cyclic AMP, activate protein kinase A, phosphorylate like crazy in the cell, bring glucose back in. It's a sequence of events. Questions? Now, before we leave this topic, it's also important to know cyclic AMP is so important that you have to turn it off. You have to have a way of turning it off. So if here's adenylate cyclase, here's ATP going into cyclic AMP. You break it down through an enzyme called phosphodiesterase, which converts it to 5' AMP. So even though you make cyclic AMP, you have to also get rid of it, because if you have too much of it, you basically can have cholera if you never got rid of it. So you can regulate this. Why do I bring this up? How many of you who had breakfast today, or didn't have breakfast, doesn't really matter, drank coffee, drank tea, or ate some chocolate? Yeah, I'm a hardcore coffee drinker myself. What you did not do is simply drink coffee or tea. What, in fact, you did was you were drinking a phosphodiesterase inhibitor. Caffeine in coffee, theophylline in tea, theobromelain in chocolate are, in fact, phosphodiesterase inhibitors. That part of the reason you get that little extra charge from drinking this stuff or eating it is, in fact, you're inhibiting this phosphodiesterase, and you keep your cyclic AMP levels up for a longer point in time. Therefore, you initiate the response for a longer point in time. Okay, so next time you go to Pete's or whatever you go, or some local place, just kick back with your coffee and say, oh, that's a good phosphodiesterase inhibitor I'm enjoying. Okay? Now, also the reason why some people respond, caffeine has 3 methyl groups, theophylline has 2 methyl groups, theobromelain in chocolate has 2 methyl groups. Each phosphodiesterase is slightly different, just ever so slightly different. That's why some individuals like coffee over tea or get a little extra charge in coffee over tea. Some people, tea doesn't do anything for you. Other people, tea does a lot, but not coffee. Some people like chocolate. Most people I know like either coffee, tea, or chocolate. It's rare that somebody likes all 3. And that's because I think it's because your phosphodiesterase reacts to each of these inhibitors in a slightly different way. Just my theory on this. Okay? Now the segue to human reproduction is going to be as following. As you can imagine, these pathways are very, very important. pharmaceutical companies love to look at these pathways and figure out how to manipulate the pathway for making new medicines. So one company had an idea that you can create a new phosphodiesterase inhibitor because they thought the possibility that it could be involved in blood pressure. You could reduce blood pressure by inhibiting phosphodiesterase. And there's some theoretical aspects to it that says that that's the case. So where would you try to treat a novel drug with a lot of people with high blood pressure, generally? Usually at an old folks home, right? If most people your age don't have hypertension drugs. Ask your parents and grandparents. I'm sure those with high blood pressure are on some, okay? So they went to an old folks home, and they tried to give these people this phosphodiesterase, this new phosphodiesterase inhibitor, this drug. It had unfortunately, after a month, it had absolutely no effect on blood pressure. And they were going to discontinue the study, and this old folks home went nuts. They said, no, we need our drug. We need it. And they're thinking, oh my gosh, maybe they developed some sort of addictive drug and everything. Well, they sorted it all out, and the name of this phosphodiesterase inhibitor is Viagra. Okay? So what's going on here? Because coffee does not have the same effects as Viagra. If it did, can you imagine the stock of Peet's and Starbucks coffee, right? It'd go way up. It'd almost be like Apple, probably greater. Well, it turns out they didn't realize at the time there are different kinds of phosphodiesterases with different kinds of specificity. This phosphodiesterase that Viagra inhibits is a cyclic GMP phosphodiesterase, converting it to 5' GMP. And what makes cyclic GMP is guanylate cyclase that converts GTP into cyclic GMP. Okay? And what does this have to do with the actions of Viagra? Well, it turns out cyclic GMP has a lot of signaling pathways involving some phosphorylation, causes muscle relaxation, arterial dilation, and as you'll see on Wednesday, that's exactly the mechanism of an erection. Okay? And that's what causes that. Now, when you have the advertisements for Viagra and related kinds of phosphodiesterase inhibitors, reproductive aids, what are some of the, if you listen to these commercials, what do they say? Take this to your doctor, blah, 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 but you might go blind, right? Do you ever see that? Like temporary blindness. Everybody hear that in the commercials? Turns out the other place that the cyclic GMP phosphodiesterase is incredibly important is the rod cells in your eyes. So when you inhibit that phosphodiesterase, you can, in fact, go temporarily blinded while you have way too high levels of cyclic GMP. And in about 20% of the people that take Viagra, they can have visual problems, temporary visual problems. So it all ties together physiologically. It's a classic example of thinking a drug does one thing and you end up with something else. Okay? A lot of the, like for example, tamoxifen, which is an anti-breast cancer drug, was originally developed as a contraceptive. Wasn't that good of a contraceptive, but it was a great anti-breast cancer drug. This happens quite a bit. Okay, questions on this? Okay, so now you can really tease your parents or grandparents about taking phosphodiesterase inhibitors next time you see them drinking coffee, for example. Okay? So, this is our segue into reproduction. Not a bad segue when you think about it, right? So, and I'm going to focus on human reproduction. I'm sure in the lab you figure out how amoebas go about stuff and everything, but we're going to stay mostly with humans. In human reproduction, it's sexual reproduction in that you've got to take two haploid gametes and you make a diploid, one diploid zygote. Okay? And that is different than the way an amoeba reproduces. Okay, so what are the gametes? So the male gamete is spermatozoa. The female gamete is actually a bit more complicated, and it's technically called an ovum, but you actually don't have a separate cell that's haploid. You eventually get an ovum nucleus, but not an ovum cell that's haploid, and you'll see that complication Wednesday or Friday. But generically, and there's different names to it, depending on where in the pathway it is, but we'll give this a generic name called eggs. So if I use the word egg, I'm not really specifying where in the developmental pathway we're talking about. But it all refers to something that could become an ovum. Now the reproductive organs, in the male it's the testes, and the female it's the ovary. And the reproductive organs, the general name, are called, collectively, they're called gonads. So if you say gonad, it means you really don't know the difference between a male and a female. Because the males are testes, the females are ovaries. Well, you know the difference, but you're not. It's saying, what did you drive to school today? Oh, I took a car. Doesn't tell you what type of car, what kind of engine, how bad the brakes are, or anything like that, right? So what are the functions of the testes and the ovaries? And there's two important functions. One is, as you can imagine, is to produce the gametes. In the case of the ovary, it's a little different, that it produces the eggs, but the ovary itself never makes the final ovum. And you'll see why that is on Wednesday or Friday. But you also make reproductive steroids. Steroids are real important in human reproduction. So for the female, the two key steroids we're going to talk about is estrogen and progesterone, E and P. For the males, which we'll start getting into in a few minutes, by the way, excuse me, for males, it's testosterone, T, and dihydrotestosterone. DHT. And everybody says, well, estrogen is the female steroid, testosterone is the male steroid, right? The reality is the male steroid is actually dihydrotestosterone. And you'll see that distinction. Secondly, all females make testosterone because, as you'll see on Wednesday, testosterone is the precursor for estrogen. So if females could not make testosterone, you also could not make estrogen. So keep that in mind. So it's a little bit of a misnomer out there as to what's a male and female hormone. So in other words, it's a bad designation. Better to simply identify what they are and what they're used for. Okay? Questions on this brief introduction? So let's talk about male reproduction the next bit of time, which is spermatogenesis, which is the production of spermatozoa. Spermatogenesis is continuous because the hormonal control mechanism is all negative feedback. Right? What's negative feedback? Keeping an entity within a constant level. If it gets too much, you modulate it down. If it gets too low, you modulate it up. Too high, modulate it down. The idea is keeping it within an acceptable window. As we talked about with stress regulation. It is continuous in males because of negative cooperativity. We'll certainly hit that on Wednesday. Where it takes place in the testes is in something called the seminiferous tubules. The seminiferous tubules in the testes, the way to think about it structurally is like a big ball of spaghetti, a big plate of spaghetti. A whole bunch of interconnecting ducts that are all intermixed with each other. Okay? And what we'll look at is what's in this seminiferous tubule. We'll do a cross-section here in a second, is the general cell types. Inside the tubule are the spermatogenic cells leading to the spermatozoa, and you have a bunch of accessory cells. Oops. And these accessory cells, which I'll put right down here, are two very important types. You've got the Leydig cells, which convert cholesterol through a bunch of steps into testosterone. Again, you need cholesterol. You can't just shut out cholesterol completely. And you have what's also called the Sertoli cells that converts testosterone into dihydrotestosterone. And it is dihydrotestosterone that activates the targets for dihydrotestosterone are the spermatogenic cells. So the critical steroid, the critical androgen to keep production of spermatozoa is not testosterone, but it is dihydrotestosterone. Very important difference. Okay, because on Wednesday we'll briefly talk about male contraceptives, and that distinction becomes very important. Okay? So let's look at a cross section. So the seminiferous tubules is this big plate of spaghetti, and we're going to look at the cross section of one tubule. So it's multiple tubules. So we're looking down in it. This is the basement membrane. Outside the basement membrane is the Leydig cells. Okay? This is the cross section of the seminiferous tubule duct. Inside are the Sertoli cells. Call these just Sert, so we can, let me just. And you have the spermatogenic cells in between the Sertoli cells. These are spermatogenic. Which are these guys. So the idea is to, in the middle of the duct, so if this room is a huge seminiferous tubule duct, the idea is right in the middle here is to get all the spermatozoa and out, not to leave them on the outside. They got to go on the inside. And there's a process that takes place from one side of the basement membrane to the other. And I've drawn these Sertoli cells as kind of touching each other. Does everybody see that? And if you get them in the last minute here, if we just look at a higher magnification of this, here's the basement membrane up here. These are the Sertoli cells. It's called the Sertoli cell bridge, which would be right here. So you got some space on the outside of the bridge. This is the basement membrane. And different processes take place on the outside of this bridge to the inside. As you go on the inside of the bridge, you get to the middle of the tubule where you want to have spermatozoa. And the mitotic mitosis takes place out here with a little bit of meiosis, and a lot of meiosis takes place on the inside. Because remember, you got to go from a diploid precursor to a haploid gamete, right? What's the difference in DNA between the two? Half, right. Haploid versus diploid. And I'll just leave you with, you start out with cells called spermatogonia. These are diploid. 46 chromosomes, 2 cDNA content. And eventually, you're going to get to a spermatozoa that's 23 chromosomes in cDNA, right? And we will pick that up on Wednesday. So as you have your coffee for lunch and so forth, just, think about differences in phosphodiesterase specificity. That's all I can say. Or your tea. See you on Wednesday.